Hi, this is Miss Howell. We're going to move on to muscle physiology, which is the second half of this unit on muscles. Remember that this time we're going to look for words in red to fill in your blanks. Use the stars to help point out things that you might need to know for the test coming up. Take your time with this. It's only a 15-minute recording, but you may stop and pause and replay as many times as you want to try to get all of your words filled in on your blanks. So we're going to go ahead and move forward. And here we go. In Module 5A, we mentioned that muscles have 4 characteristics. One of those was contractility. If you look back in your notes, you'll see that list. Muscles cause movement because they contract. The way in which they contract is called the sliding filament model. Strands of skeletal and cardiac muscles have striations or stripes on them. The pattern of stripes is created by repeating units of sarcomeres within the myofibril or muscle fiber. So here's a diagram similar to the one in your book. Muscle or meat is a high protein food. 2 important proteins within your muscles are myosin and actin. Of these 2, myosin is thicker. Contraction is caused within a sarcomere, which is 1 unit, when in unison all of these myosin heads grasp the thin active actin filament, that's these blue in the picture, and they pull on it, shortening the sarcomere. So if you look here, you see these little projections, these little knobs that come out of the myosin. They grab the actin, and all in unison they'll pull at the same time. Thick dark stripes are called A-bands. Oops, excuse me. A-band. So A-band is listed here. That's these dark stripes if we look at this in under a microscope. The lighter area, when you're looking at the slide under a microscope, are called I-bands. That's this area here, because these dark thick filaments are not present. These separate their outer boundary is called a Z-line, or sometimes a Z-disc, or sometimes a Z-band. But when you see the letter Z, that's the outer boundary from one sarcomere to another sarcomere. So that's 1 unit. These separate one sarcomere from another. And finally, there's the H-zone, which is the area between the ends of the actin myofilaments, the blue to the blue. So the H-zone separates 2 sets of actin myofilaments. All right, so get that picture in your mind, and I'm going to move on and explain how this contraction works. When a muscle contracts, the sarcomere shortens, pulling the Z discs closer together. So go back and look at the diagram and notice that when it contracts, both the H-zone and the I-band are shortened. This shifting of the actin by the myosin to shorten the sarcomere during this contraction does not change the shape or length of the myofilaments that do all the work. The size of the actin and the myosin remain the same. Note the star. And because the A-band is the same as the myosin, it's also unchanged. The important thing for you to understand about muscle contraction is that the presence of calcium ions, which have diffused out from the sarcoplasmic reticulum, cause accessory molecules, which are troponin and tropomyosin, of the actin filament to change position, exposing the active site that attracts the myosin head to bind and pull. After the pull or contraction, the nerve stops sending its signals, and the sarcoplasmic reticulum actively gathers up all the calcium, hiding the active sites again, and the muscle fibers then relax. We're going to watch the Hank Green video, and it'll have some animation that kind of shows that. It makes more sense. Energy needed for the work of myosin heads to grab the active site of the actin filaments is supplied by ATP. ATP is changed to ADP and phosphorus when it releases energy for use. As the energized myosin head binds to actin, the remaining ADP is dropped, and it pulls the actin to shorten the sarcomere, causing contractions. This is called the power stroke. A new ATP molecule then binds to the myosin head, causing it to release the active site. This new ATP then breaks down releasing more energy, allowing the head to spring back, ready to work again. This ATP-driven spring back is termed the return stroke. A neuron is a nerve cell. Muscles are controlled by nerves, which are made up of motor neurons, which are cells that are specifically designed to carry signals from the brain and the spinal cord to skeletal muscles at a very high velocity. We'll study neurons in a later chapter, but just for now, I want you to know that each motor neuron has a long process, which is called an axon, and this extends from the main body of the nerve cell. The axon is what carries the nerve signal. When the axon of a motor neuron reaches the paramycium of a bundle of muscle fibers, remember the paramycium is that outer layer of the bundle of fibers, it forms a connection called a neuromuscular junction. Oddly enough, no nerve in the body actually touches the muscle that it stimulates. There's never an actual touching connection. There's a very, very small gap between the end of the axon and the muscle fiber called the synaptic cleft or a synapse. The communication between the nerve and the muscle occurs across this gap or across the synapse. Acetylcholine, which is abbreviated, thank goodness, ACH, is a neurotransmitter. These are chemicals released by the neuron, allowing it to communicate with other cells. In this case, the ACH interacts with the skeletal muscle fiber, and it creates a signal that ultimately causes the cell to contract. The body makes an enzyme to get rid of ACH, thus allowing your cell to relax. This enzyme has a big long name called acetylcholinesterase, and it is essential. Without acetylcholinesterase to destroy ACH, a muscle cell would stay contracted maybe forever. The whole process starts with an action potential, which travels down the axon of a motor neuron like an electrical message. A presynaptic vesicle then moves to the membrane and secretes ACH. The ACH action potential travels across the synaptic cleft and is taken up by the muscle's fibers postsynaptic membrane. The message or action potential travels down the muscle fiber membrane to a T-tubule pit in the sarcolemma. The ACH action potential enters the sarcoplasmic reticulum through one of those T-tubule pits. The sarcoplasmic reticulum is full of calcium ions that have been brought in to this area via active transport. The action potential impulse within the sarcoplasmic reticulum stimulates the diffusion of calcium out so that the calcium can be picked up by the myofibrils or the muscle cells. Recall that once in the muscle cell, the calcium ions that are present on actin filaments expose the active sites, which attracts the myosin head to latch on and contract the cell. There's several stars on this slide and the one before, so go back and make sure you understand those. The first thing that must happen to stop a muscle from contracting is the nervous system, the brain and spinal cord, must stop sending the action potential down the neuron. As soon as the action potential is stopped, then ACH is no longer released from those presynaptic vesicles. No more ACH is there to travel across the synapse. Any residual ACH that's in the muscle begins to be broken down by that enzyme we mentioned, the acetylcholinesterase, which was produced by the muscle fibers themselves. Now that the muscle action potential's gone, the calcium ions are no longer being diffused out of the sarcoplasmic reticulum to help out with exposing the active site of the actin filaments. These sites then close off. Cross bridges between actin and myosin can no longer form. The sarcoplasmic reticulum returns to its previous role of actively taking in calcium ions to store them for the later use when the whole process is going to start all over again with another muscle contraction. Have you ever heard of rigor mortis? Rigor mortis is a short-term stiffness that occurs after death. This stiffness does not last forever, however. Why? Well, remember that calcium ions are gathered back up into the sarcoplasmic reticulum by active transport. This requires energy that's supplied by ATP. Well, after death, you no longer produce ATP, so active transport stops throughout your body. Calcium then will leak out of the sarcoplasmic reticulum after death, and it will cause these cross bridges between the myosin and the actin, and the contractions will occur. That's rigor mortis. This contraction becomes stuck because the calcium can't be returned back into the sarcoplasmic reticulum until the body begins to decay. And at that point, the muscle tissues are degenerating and they just kind of decay and fall apart, and they relax at that point. Similarly, while you're still living, your muscles can also cramp up and get tight and become sore when limited ATP is produced in your body. So after death, obviously you don't make any. Well, there are times where ATP is not being made in very high supply. It's created most easily when oxygen is in abundant supply via aerobic cellular respiration, which hopefully you learned last semester. Low levels of oxygen put your body in a state of anaerobic respiration. ATP is created still, but in much lower amounts. This can cause your muscles to still contract, but they can remain tight and cramped because not enough ATP is available to release those myosin heads from the actin filaments and allow the muscle fibers to then relax. So this anaerobic respiration leads to lactic acid as a byproduct. Here's some terms you need to know. Motor unit. A motor unit is one motor neuron, remember a nerve cell, and all the muscle fibers that it innervates. And to innervate means to control. So the axon of the motor unit connects to the cell at the synapse, but remember they don't actually touch. Excuse me. Depending on the motor neuron, it may branch only a few times, or it may branch hundreds of times from one single motor neuron. Why is this? Well, when power is required of a muscle over, say, precision, there are only a few very large motor units at work. But when precision is key for fine motor control or delicate movements, very small motor units are at work. Every motor unit produces a single action potential that travels down the axon, but maybe to many muscle fibers. In a given motor unit, then notice the star, all of the muscle fibers will contract to exactly the same extent with every action potential that is supplied by that single motor unit. Each motor unit will follow the all or none rule, meaning they contract fully or not at all. However, each muscle is controlled by many motor units. Being served by many motor units is what allows you to flex your bicep muscles only partly and not all the way every single time. In addition, the strength of the nerve stimulus also plays a role in the power of the muscle contraction and response. So let's talk about the nerve stimulus for just a moment, then we'll be finished. There's a sub-threshold stimulus, which is one that's too small to create an action potential in the neuron at all. A sub-maximal stimuli of increasing strength creates more action potentials along more neurons. You get more, but not all. And then maximal, which is a stimulus which is strong enough to create action potentials in all the motor neurons, innervating your whole muscle. Hopefully this will help. We'll talk about a lot of this in class, and if you have any questions, bring them with you, or you can send me text messages, and I'll do my best to answer you through the week. We'll see you in class.